figure something out. I'm not exactly sure what, but I would.
What would you do for me, Mr.
I don't know.
You can't mess with my name. I'll play your full name down.
All right. So this should all be review for you guys. I know we've learned this, but you need to sort of refresh your memory about variables and control groups and so forth. Okay. So first of all, what is science? Well, you can sort of think about science as two things. It's sort of a whole bunch of stuff we know, right? I gave you that big, thick textbook. It's filled with facts and information about living things. So science is a whole bunch of stuff we know. But science is also a way of finding out new things, a philosophy about how we can learn new information. When we talk about the scientific method, that's what we're referring to. So how do scientists find out information? How do they determine all those facts in that textbook? They did that by following a certain process. It's a way to answer questions about the natural world. And you know, I know in Southern Care we learned people really hate elephants on cars. And that's one way to think about it. But there's not one scientific method. There isn't one single set of steps, really, that an actual scientist follows. It varies. But all scientists formulate hypotheses and test them and so forth. Okay? We sort of put them in a framework to talk about them, but it can happen in many different ways. So how do scientists learn about the world? Well, you follow this little flow chart. Starts with asking questions. So asking a question about the natural, asking a question that we don't know the answer to. That's where you start. And as you know, before you actually set up your experiment, you come up with your hypothesis, what you think, your prediction. And that's what you're testing in an experiment, your hypothesis. And you set up an experiment that can test that hypothesis. You look at the results of your experiment, and you compare the results to what you predicted. And you make your conclusion. You may conclude, yeah, I was right, my hypothesis was correct, or maybe it wasn't correct. Your hypothesis is correct, and you could ask a new question. If it's incorrect, maybe you come up with another hypothesis to test. And this process repeats time after time, and that's how we gather these facts, these scientific facts that build up over time to create larger theories. So if we imagine our steps that we could use as our example, in the problem step, we ask a question. So the scientific method is how we acquire new knowledge. So we ask a question. Now, it can't just be any question. You can't say, what is the meaning of life? Why am I here on this earth? Those are not questions that I could answer using science. Those are philosophical questions.
I have a question.
Scientific questions are questions that we can set up an experiment and we can answer. We can find the answer to these questions. So, some examples. How does the amount of nitrogen fertilizer affect the height of pea plants? Does increasing the temperature of an enzyme increase its rate of reaction? Do high doses of vitamin C prevent people from getting the common cold? All questions that we could answer. We could set up an experiment. You could probably envision some of these experiments, and we could get some information and answer some of those questions. But before we go about doing that, we need to have some background knowledge. Okay, we need to know a little bit before we can actually set up our experiment. Maybe before we can come up with our own hypothesis, we need some background knowledge, so we do some research. In the experiments I just mentioned and the questions I asked, maybe we need to know sort of, okay, well, what generally do pea plants require? What kind of nutrients? Or what are the enzymes I may be able to use if I'm setting up this enzyme experiment? What's the recommended dosage of vitamin C from the FDA? So I might want to know those things before I set my experiment up. So the research phase, we're not sort of doing our experiment. We're just gathering background. Once we've done that, we can start with our hypothesis. This is one of the most important steps. Coming up with your hypothesis. It's your predictions, what you think the outcome of your experiment will be. Before you actually complete your experiment, you predict what's going to happen. And there's some important points about formulating your hypothesis. It must be something testable, and it must be specific. I can't say nitrogen will help pea plants grow. It's not specific enough. What does grow mean? What does help mean? Okay? I need to have specific something. When I run my experiment, I'm going to be able to easily say yes or no, supported or not supported. Often, it's best to write your hypothesis as an if-then statement. Meaning, say, if I do this to the independent variable, then I predict this will happen to the dependent variable. Okay? If you phrase your hypothesis in that way, it's easy to figure out what's the independent variable, what's the dependent variable. If I change this, then I think this is going to happen. So some examples. If pea plants are given additional nitrogen, then they will grow taller. Again, I have my independent variable, the amount of nitrogen, my dependent variable, the height of the plants. If the temperature of salivary amylase is increased to 40 degrees Celsius, then enzyme activity will decrease. Independent variable is temperature, dependent variable is enzyme activity. If individuals are given 200% of the RDA of vitamin C, then they will be less likely to contract the common cold. Do you have the whole thing? Yeah. Okay. Oh, no, you don't. So add that in. Contract common cold. Got cut off. So it doesn't necessarily, you can have a correct hypothesis that's not an if-then, but it's often helpful to phrase it that way. So those are all statements. Remember, hypothesis is never a question, it's a statement, a prediction. And they are testable. They're specific. You could probably tell me right now what sort of experiments I would do that would test these hypotheses and give me a pretty clear answer as to whether they're correct or incorrect. That's what we do in the experiment. Basically, it's just a procedure that's going to test our hypothesis. In science, we use controlled experiments. Experiments where we carefully monitor and adjust different variables. We always have multiple groups in our experiments, a control group and one or more experimental groups. Control group is the one we need as a comparison, as sort of a baseline. In the control group, we're not altering anything. We're not changing the independent variable. It's under normal conditions. The experimental groups, we change the independent variable. We're altering something. They're getting a treatment. Something about them is being changed. For example, the pea plants that would be given just regular water would be a control group. Pea plants that are given additional nitrogen along with that water would be an experimental group. And I could have multiple experimental groups. Maybe some get 10 grams additional nitrogen. Another group gets 20 grams and 30 grams. So I may have several experimental groups in my experiment. That's okay. What? Sorry, it's off a little bit. So, control group is treated normally. Experimental group is the group that has the independent variable changed.
that is have the independent variable change.
The group or groups that have the
But it
independent
says
variable
that
change.
is.
That have
Okay.
the independent variable changed. So, let's talk about these variables. Independent variable. In a good experiment, there is only one of these. Remember, in a controlled experiment, almost everything should be the same between your groups. Ideally, you want just one single thing to be different. Now, in reality, it's tough to control every single variable. But we do the best that we can to be sure there's only one independent variable. So in my pH experiment, it's the amount of nitrogen. In the enzyme experiment, it's the temperature. In the cold experiment, it's the amount of vitamin C. Now, often when we ask for the independent variable on a quiz, people give me too much. People start giving the problem. Will vitamin C help people get better? Okay? The question you need to answer, if I ask you what is the independent variable, you're going to say, okay, what was different between the groups? The difference was the amount of nitrogen. The difference between my enzymes was what temperature they're at. The difference was how much vitamin C they got. So just be succinct in your answer in those questions. The dependent variable, this is the data you collect, the information you're gathering, is what you're looking at when you make your conclusion, such as the height of the pea plants, the amount of enzyme activity, the number of people that get a cold. When I ask these questions, often people are giving me results, but that's not what I need. When I ask for the dependent variable, you're telling me, what are we measuring? You're not telling me that the plants with greater nitrogen grew taller. You're just telling me, what did we measure? And then we have our variables that we're trying to control, variables we're holding constant. There are many of these. There's lots of things we try to equalize between our control and experimental group, such as all this type of soil, the amount of water, the type of pea plant we use, the pH of the enzyme solution, the age, diet, and general health of my test subjects in the vitamin C experiment. So we want to try to equalize as many of those variables as possible so that the only difference is the independent variable. So, independent variable is the factor, the variable you're changing. Dependent variable is the data that is being collected and variables held constant are the factors that are the same between each group. So I started making these notes this summer in Google Docs. And this is only the second one that this might be the first one I did. I got better in your next one, so don't worry. But need to adjust some things.
I say we do vitamin C, and Chloe and I can drink orange juice all day.
Yeah.
I love orange juice. Better than?
What?
Excuse me?
What's it say?
Factors that are kept the same between the groups.
Oh.
Wait, is that supposed to be upward variable?
Yeah, that's
held
just
constant?
with variable held constant.
I wasn't.
I know you're not.
Then why did you change it?
Oh wait, I was gonna
I'm so tired.
Alright, let's finish up this next slide.
Like I just
We still have 2 minutes. Not time to put things away, Linda. So, observations. This is when you collect your data, when you measure the dependent variable. Sometimes you're doing that as the experiment's going along. Other times you do that at the end of your experiment. Depends on your experimental design. Sometimes our observations are quantitative. Most often they're quantitative. They have a number with them. They're measured. Other times it could be qualitative, the color of the leaves, for example. We often put this data, these observations, into graphs or tables or charts so that we can visualize it better. Some examples. We measure the pea plant's height every 3 days. Or we measure the amount of gas produced in the enzyme reaction. Or we see how many people contracted a cold during our experiment. Those are examples of our observations, our data collection. And then our final step is to make a conclusion. We look at our data and we say, okay, was my hypothesis correct or was it incorrect? We compare the control group data to the experimental group's data. And this is an important fact, this third bullet. Something we can now start to think about because you guys are getting more advanced here in science. And that a difference between the two groups, the control and experimental group, has to be what we call a statistically significant difference. Sometimes there are small differences that may just be due to random chance. But we have statistical ways to tell if it's a significant difference. It's a difference enough that we're confident.
Is the tool quiz tomorrow?
Yeah, tool quiz tomorrow. Estudiar. That means to study.
Is this still part of like